like to welcome Dr. Stephanie Hanna. She's a DRWF research fellow. This is a UK-based 3-year research fellowship coming from the Diabetes Research and Wellness Foundation. And we're very happy to have her discuss her work. Intradermal injection of diabetes autoantigens results in recruitment of clonally expanded antigen-specific T cells to skin and draining lymph nodes and facilitates monitoring of autoimmune response. Dr. Hanna is currently, as I mentioned, the DRWF Professor David Matthews Research Fellow at Cardiff University. She completed her undergraduate degree and PhD in pharmacology at the University of Bath with placements at Novartis and Pyramid. Her research interests focus on the immunology of type 1 diabetes using cutting-edge single-cell sequencing and bioinformatics to analyze antigen-specific responses and lymphocyte subsets in response to diabetes autoantigen challenge and immunotherapies. In addition to her DRWF fellowship, she is also a co-investigator on a Helmsley Charitable Trust funded experimental medicine study to define the efficacy and mechanisms of peptide immunotherapy for type 1 diabetes. Welcome, Stephanie. Thank you very much for joining us. We're really interested to hear what you have to present today.
Oh, well, thank you ever so much for inviting me. So today I'm going to talk you through some of our work on intradermal challenge with self-antigen and how I've been combining that with single-cell RNA sequencing to profile these autoreactive T cells in type 1 diabetes. So I expect for this audience, this doesn't need an introduction, but type 1 diabetes is an autoimmune mediated destruction of the insulin-producing beta cells in the pancreas. And autoantigens can include things such as proinsulin, GAD, glutamic acid decarboxylase, which I'll be talking a lot about today, and IA2, as well as other proteins, hybrid insulin proteins, for example. And those diabetes autoantigen specific T cells are enriched but not accessible in the living human pancreas, and very few of them circulate in the peripheral blood. So we can be talking about 1 in 10,000 T cells being specific for a given autoantigen. And that's a real barrier to being able to identify biomarkers for monitoring disease activity and for developing new immunotherapies. So what I'll be talking to you about today is my approach of using intradermal injection of GAD to relocate these T cells to easy to sample locations and to characterize them using single-cell RNA sequencing. And at the end, I'll also touch upon how we've been using an activation-induced marker assay to find these cells in the peripheral blood too.
So in essence, you're kind of luring them to a site where you can, capture them and then analyze them. So it's really unique. It's really great.
Yeah. So this was an approach that was developed by Daniela Tatarik, who's a clinical research fellow in our group. And sort of her hypothesis was that here's the pancreas and these, which will be enriched with those autoantigen specific T cells in the pancreas and the pancreatic lymph node. And if she injected a pancreatic protein into the skin, that would facilitate the trafficking of those T cells to the skin draining lymph node where they could interact with their cognate antigen. And from where she would be able to sample them using ultrasound guided fine needle aspiration of the lymph node. But what we also observed was the delayed appearance of a red mark at the site of injection. And that suggests to us that those T cells might also be homing directly to the site of injection. So from there, we were able to sample them by raising a suction blister over the injection site and taking the blister fluid, which contains the T cells. And just to say at this point that in our hands, these are quite routine procedures that are reported as painless, and we've performed in several dozen volunteers to date. So once we have those samples from the lymph node and from the blister fluid, what I've been doing is single-cell RNA sequencing, and I just wanted to briefly introduce it in case people weren't familiar. But we can think of bulk RNA sequencing as being like this fruit smoothie, where you have the transcript of every single gene in each cell, but everything is jumbled up together, so you can't tell the phenotypes of any of the individual cells. Or you can think of flow cytometry as being like this cartoon fruit bowl, where now we're gaining information about individual cells, but only a few pieces of information at the protein level for each cell. Whereas what single-cell RNA sequencing allows us to do is like this very high definition fruit salad, where you've got the expression of every single gene in each cell. So we can be talking about 5,000 different genes expressed in each cell. And I also use the 10x genomics platform, which allows me to get the T-cell receptor sequences for each cell as well. And then what I'll show you on the next few slides will be some UMAP plots of this single cell sequencing. And just if you're not familiar with the UMAP plots, what that does is it takes the gene expression of all those thousands of different genes in each cell, clusters similar cells together, and then projects them onto a two-dimensional space. So here, each of these little tiny dots is an individual cell with similar cells, such as all the mast cells clustered together. So what I've also been able to do with the T-cell receptor sequences, so those, if I just briefly introduce them, so there are billions of possible T-cell receptor sequences, and some of those TCRs will recognize diabetes autoantigens. So here you can see the autoantigen in the context of an MHC. And when that TCR recognizes the autoantigen, it will trigger that T cell to destroy the beta cell. But not only that, when a T cell is activated through its TCR, often what happens is the T cell will proliferate, and it makes what we call clones, because these T cells here all bear identical TCRs. So where we see that happening, we know that hasn't happened by chance because there are so many billions of possible TCRs. This has happened because T cells interacted with its cognate antigen and proliferated. So here where we see 3 cells bearing the same TCR, we give that a clonal number of 3. And this work really started from a clinical trial giving an intradermal injection of proinsulin peptide C19-A3 conjugated to gold nanoparticles. And what was observed during the clinical trial was there's persistent redness and induration at the site of injection, and that could persist months and even years after injection, which for me as a basic scientist was a very interesting opportunity to understand the immune process. So what we did was we took suction blisters and also some biopsies of the injection sites, and I was able to study the T cells. And what we saw here in the blister fluid were large numbers of T cells. So here we have lots of infiltrating CD8 T cells, with mostly with resident memory and effector memory phenotypes, and also memory CD4 cells and Tregs. And I can also look for those clonally expanded cells, thinking that those are the ones that are most likely to be antigen specific. And you can see I had very large expansions of, I've lost my cursor, very large expansions of CD8 T cells, but also of those memory CD4 T cells as well. But we really wanted to know what antigens they were recognizing. So we collaborated with Maki Nakayama to use her 5kc model. So this is a cell line where you can re-express your TCR sequence that you've obtained from your single cell RNA sequencing. And here where the TCR interacts with its cognate antigen, it will activate the 5kc to produce IL-2. And in that way, we can determine the specificity of the TCR. And when I mapped the TCRs back onto the UMAP plots, you can see the phenotypes of the cells. So here you can see my pro-insulin specific T cells that are both CD4s and CD8s, and very large clonal expansions of gold nanoparticle reactive T cells as well as some T cells which were not specific for either antigen in our hands. But that was very exciting to us because now we'd been able to identify some of these clonally expanded pro-insulin specific T cells that were resident in the skin months after that injection of proinsulin peptide. And we also have their complete gene expression profile. So we're able to look at what makes those proinsulin specific cells unique compared to the other infiltrating T cells. And so, for example, here you can see those proinsulin specific cells enriched in KIR ligands and also in granulysin compared to those that are either gold nanoparticle specific or non-specific. So that was quite interesting for us, because what we were able to demonstrate with that clinical trial was that intradermal injection results in those clonally expanded antigen specific T cells in the skin, and they persist there for months to years, and they got that spectrum of tissue resident memory and effector memory phenotypes. And so that was interesting for two reasons, really. That one, it was quite a novel mechanism of generating tissue resident memory cells in response to vaccination, if that's what you're interested in. But for us as diabetes immunologists, as a route to being able to monitor the diabetes autoantigen specific cells that are so rare in the peripheral blood. So that preprints up on medRxiv now. But the question that I really wanted to ask was, could we use a single injection of the diabetes autoantigen to recruit antigen-specific T cells? And to do this, we use GAD alum from diamide. So to start off with, back into the blisters. So here are 3 control blisters from people with diabetes who had not received an injection of GAD alum. And you can see there's low numbers of total T cells in the blister fluid, and they have low clonal frequencies. Most of them have a clonal frequency of 1, so each cell has its unique TCR. But when we injected people with GAD alum intradermally, we do see this delayed appearance of a red mark at the site of injection. But with the GAD alum, unlike the gold nanoparticle C19-A3, this is a very, very transient appearance of a red mark. But nonetheless, we'd set this up, Daniela had set this up so that we could capture those cells at the exact point that they were in the skin. And so we're looking for those clonally expanded cells because we believe that they're going to be the ones that are enriched in the autoantigen specific cells. And here you can see that in these 3 participants who'd received an injection of GAD alum, we have significantly more total T cells in that blister fluid. And we have this big increase in the clonally expanded cells, so up to 15 cells bearing identical TCRs. So that was very exciting and suggesting that what we're detecting is an antigen-specific infiltration occurring. But for these participants, we were also able to get the fine needle aspirations of the lymph nodes and do a single-cell RNA sequencing from those. So here again, I've shown all the cells from the lymph nodes. So we got clusters of B cells and also of T cells. And from those, I can map onto them the T cell receptors, and specifically what I was interested in was those clonally expanded TCRs here in the spectrum from green to yellow to red. And you can see clonal expansions both in the CD8 subset and in the CD4s. So we were working on a hypothesis in the post-injection lymph node that those would be enriched in GAD-specific T cells. So we went back to Maki Nakayama's 5KC model, and this time we are presenting GAD to this TCR to see whether it recognizes it and triggers the 5KC to produce IL-2. And here, for example, you've got a positive control of anti-CD3, CD28 stimulation and a negative control, but you can see that of just the antigen-presenting cell with the 5KC alone. But when you can see that when you give that antigen-presenting cell exogenous GAD to present, or when we use the lentivirus to drive endogenous expression of GAD in the APC, that's recognized by the TCR, and the 5KC cell produces IL-2. So what I was able to determine from that is that those antigen-specific cells comprise around 30% of the clonally expanded T cells in both the lymph node, the post-injection lymph node, and in the blisters, which is just a massive enrichment compared to that 1 in 10,000 cells that are present in the peripheral blood. Because I have the complete gene expression of all of those cells, I can start to compare what make in the lymph nodes where there are clonally expanded cells present pre-injection, but then compare those to what's present post-injection of GAD, where we know we have the GAD-specific T cells. And when we look at those differentially expressed gene pathways, what we can see is that those post-GAD expanded cells have a highly activated phenotype compared to the clones present pre-GAD. So as you would expect, pathways involved in TCR recognition of antigen because we've introduced their cognate antigen, but also pathways such as cytokine signaling in the immune system, and also cell death and apoptosis signaling pathways. And we can also look at that another way. You can look for your favorite genes and how they're differentially expressed. So here's the top 30 differentially expressed genes between those clonally expanded cells pre and post injection. And so, for example, here you can see genes involved in chemotaxis such as CXCR4 and EZR, and also other things such as TXNIT, which is regulated by glucose and insulin, and is downstream of verapamil. So if I just sort of sum up at this point where we got to, what we've found so far is that intradermal injection of GAD resulted in the recruitment of antigen-specific T cells to accessible sites in the skin and the skin draining lymph node. And hopefully what I've shown you is that those phenotypes that are clonally expanded and antigen-specific T cells in the lymph node post-GAD can be used to identify the novel biomarkers of autoreactor cells that we believe can be used to monitor immunotherapy effectiveness and tolerance induction. But we also wanted to know whether we could walk backwards into the peripheral blood. And to do this, I went to Tim Tree's lab at KCL to use his activation-induced marker assay. So here I have my same participants pre or post injection of GAD-alum in vivo. And I take the peripheral blood samples and isolate the PBMCs from both time points. But then all of those PBMCs undergo a further 18-hour in vitro restimulation. So they're either unstimulated, from which we sorted the unactivated CD4 cells, or they were stimulated with a control buffer, or with GAD itself, or with SEB as a superantigen as a positive control. And from these three conditions, I sorted the activated cells, so either activated effector T cells or activated T regs. And then all of these sorted cells we subjected to single cell RNA sequencing. And what we found was that activated GAD-specific cells had a really unique phenotype. So here's the UMAP plots of all of the sorted CD4 T cells. So we had 11 different clusters. And you can see here is our positive control of SEB stimulated ones, and then these unstimulated ones with their very distinctive phenotype. But going forwards, what I'm going to be comparing to is this control stimulation with the buffer. So these are either cells that have been activated in response to buffer components, or were already activated at the point at which they were extracted from the person, or these ones that are activated in response to GAD stimulation, which we presume to be GAD-specific T cells. So we're comparing now these two groups pre and post the in vivo injection of GAD. And what we really found from that was those GAD-specific cells, but not control cells, change phenotype upon in vivo exposure to GAD. So here on the left, we have those control activated cells, pre in vivo GAD and post in vivo GAD, with no significant changes between the cells falling into all of those 11 different clusters that I showed you on the previous slide. But here with the GAD-specific cells, pre or post in vivo injection of GAD, you can see these three clusters that change in abundance upon in vivo exposure to GAD. And what this boils down to is that pre-injection of GAD, this cluster 4, is much larger compared to the control activated cells, and is a very unique population of IL-22 positive, IL-32 positive, CD26 positive and LTB positive cells, which is largely lost upon in vivo exposure to GAD. We see that change in phenotype of those GAD reactive cells occurring. So if we look at that another way, here are my 11 clusters, and you can see here that this cluster 4, which is abundant in the GAD-specific cells pre in vivo exposure to GAD, is enriched not only in the IL-22 and IL-32, but also in other cytokines such as IL-17 and interferon gamma, and there's also an activated population you can see here in terms of CD69 expressional cells, so CD62L. And if I show you that change in in phenotype that happens upon in vivo exposure to GAD. Again, you can track here the top 30 differentially expressed genes. So, for example, you can look for your favorite ones, such as IL-32 or IL-22, and other ones such as FAS and DPP4, which is the gene encoding CD26. Or we can look at we can cluster those differentially expressed genes into pathways and look at how those GAD-specific cells change phenotype. And again, you can see the same. You can see similar patterns occurring. So you can see TCR signaling, but you can also see interferon signaling, cytokine signaling, and regulation of adaptive immune responses and programmed cell death pathways that are differentially expressed upon a single in vivo injection of GAD in those GAD-specific cells. So I hope what I've shown you there is that, in addition to my previous findings, that we can track that phenotype of the GAD-specific cells in the peripheral blood and how it changes upon that single in vivo injection of GAD alum. And we wanted to take this forward as far as possible. So with collaborators at the University of Birmingham, we have a grant from the Helmsley Charitable Trust to enable us to take this forward towards monitoring immunotherapies. And in this grant, what we'll be doing is tolerizing participants with escalating doses of a subcutaneous GAD peptide or a saline control, and then challenging with intradermal GAD peptide. And then those participants will be giving lymph node and blister aspirate samples and PBMCs to perform AIM assays. And we'll really be looking for clonal expansions and evidence of tolerance in those who've had the escalating doses of GAD peptide. So how we set about identifying which GAD epitopes to use for those peptides was done in collaboration with David Wraith at the University of Birmingham. So what he's done is he's screened both healthy controls and people with type 1 diabetes using short 30-mer GAD peptides. And he's been able to identify these 30-mer GAD peptides, which are frequently recognized by T cells in both healthy controls and people with type 1 diabetes, which is necessary to be able to start tolerance induction. So, for example, if we take this P5 peptide, what we've been able to do is take that and use it to screen through our known GAD reactive TCRs from the blisters and the lymph nodes. And here, for example, we have P5. So this is a GAD reactive TCR from a post-GAD lymph node responding to a GAD expressing EBV line or a CD3 positive control, but also to overlapping 15-mer peptides from that P5. So that's how we set up this study to identify those GAD peptides that we'll be using. So if I just wrap that up all together, I hope what I've shown you in the last quarter of an hour or so is that how we've been able to use that intradermal injection of GAD to recruit antigen-specific T cells to accessible sites in the skin and the skin-draining lymph node, and how we can use those phenotypes, especially the clonally expanded antigen-specific T cells in the lymph node, to be able to identify novel biomarkers of autoreactor cells that we hope can be used to monitor immunotherapy effectiveness and tolerance induction. And indeed using the AIM assay, that phenotype of the GAD-specific cells in the peripheral blood can be monitored and changes upon a single in vivo injection of GAD alum. And that we really think this is a route to offering people the most appropriate immunotherapies and clinical trials. So just like to wrap up with my acknowledgements, particularly to the DRWF who funded my fellowship.
Thank you, Stephanie. I mean, wow, this is just a very innovative and creative scientific approach, and congratulations to you on a very, collaborative and detailed analysis. There's a lot of work here behind what you've shown, and it's fascinating. I just want to comment before we get to questions, which we have a few.
Sure.
As we've been, we initiated and facilitated the build of a T1D TCR repository here in the US with a group in Canada, and Maki Nakayama, Todd Brusco, and Aaron Michaels were some of the early contributors to that. Of course, Maki has some very detailed data sets, and it's just been amazing to welcome her into the repository. But it seems that you all are generating some very interesting data as well, very interesting TCR data as well. And we'd be happy to have you participate in maybe one of the working meetings just to learn more and see if it's something you'd like to participate in. So I just want to put that out there because the data is beautiful. It's really great.
Thank you.
We have a question from Andrew Reeves. How long after diagnosis of diabetes were patients immunized with intradermal antigens?
So with the GAD alum, they were generally people with long-standing diabetes, so over a year and usually over 2 years.
Okay.
And then
oh, sorry.
Well, I was just going to say, and then the C19A3 gold nanoparticle was slightly closer to diagnosis.
Okay, great. And then from Sergey Pustinokov, would you expect any medical risk for the patient after expansion of autoimmunity related T cells? Did you try to stimulate the Treg response? I guess he means simultaneously.
So the GAD alum was fundiamide, so that has a very extensive safety profile. So that was why we picked that one, because we knew from their safety data that there wasn't a risk of inducing adverse effects, and these people had long-standing type 1 diabetes, and to be included in that trial they also had to have GAD autoantibodies. So we weren't risking inducing further GAD autoimmunity in people that maybe didn't have it. The proinsulin C19-A3 trial was stopped on safety grounds because of the gold hypersensitivity, which Daniela described in a separate paper. So we believe that that persistent appearance of the red mark was due to the presence of the gold nanoparticles in that study.
Yeah, I just wanted to, Stephanie has worked really hard at this, and this is something, you will, and the others will know, that we've been looking for something like this for the last 30 years. People have been very interested to say, well, who are the cells that are going after these antigens? Can we know what they look like? Because, forewarned is forearmed. If you can identify them, hopefully you might be able to target them. And I think this is really opening the way to beginning to really drill down into the fact that we do know that who some of these cells are, or we might be able to find out. And if we can expand on this phenotype to say they do look different, that will allow very much to be able to move in terms of thinking about very specific looking for things, and both not only the techniques of being able to sample potentially lymph nodes near the place, but also as a screening method to go into the blood. So we're very excited by Steph's work. I think it's really, really great. And for those of us who've been chasing this thing for many years, it is very exciting.
Yeah, it is. And she's applying all these, very newest tools to this question as well. I mean, as we know in the field, right, some people can have GAD autoantibodies and then never progress to type 1 diabetes. And in fact, a large, part of the population has these GAD antibodies and does not progress. So to be able to really finally dissect using the newest tools, which one of these, what the phenotype really is that does progress is just phenomenal. It's really great. Here's another question from Dr. Sarah Richardson. Are we being able to multiplex in tissue? We should hopefully be able to better characterize, for example, in the target organ. This should help us identify good targets to go after. Do you want to comment on that?
Yes, it would be amazing to be able to look in the pancreas and the pancreatic lymph node as well, and that's really exciting what's coming out of the NPOD as well, because that really gets that really gets to the heart of the question of which are the relevant autoantigen specific T cells. Are they the ones that are exclusively circulating between the pancreatic lymph node and the pancreas? And am I pulling them out by injecting exogenous GAD, or are all of the autoantigen specific cells that we can track in the peripheral blood with tetramers or with AIM assays, are they also the very relevant ones as well? And we're still not quite able to answer that definitively yet, I don't think.
Have you guys thought about injecting or creating these blisters in proximity to the pancreas? I mean, I saw the arm site, in those that already have type 1, have you thought about, perhaps, I mean, obviously it's deep in the abdomen, but maybe in the abdominal area?
Yeah, we haven't thought about that, to be honest, because we were keen to have the lymph nodes as well and to know where the lymph nodes were draining to. But as we become more confident with the blisters and you can see those big clonal expansions, it would be really, really interesting, wouldn't it, to see if it's easier to get those really pathogenic T cells from the pancreas and the pancreatic lymph node to home back to an injection site if it's closer to them. Yeah,
I just wanted to also ask, what does the nod data suggest? Is it similar to what you're seeing, or has anyone dipped into that model system?
I don't know anybody. I mean, obviously I've seen Andrea Schaitinger's wonderful paper about looking at which T cells are the pathogenic T cells in the NOD mouse system, and about the specific transcription factors that control whether they're staying in the pancreatic lymph node and seeding into the pancreas. But I'm not sure that anybody's tried the parallel experiment of then trying to extract them to a peripheral site by the intradermal injection. When we've done intradermal injections in NOD mice in the work that led up to that C19-A3 gold nanoparticle project in the human clinical trials, when we did do that, we could see proliferation happening in response to the intradermally injected antigens in the NOD mice. So that it was challenging to do a true intradermal injection in a NOD mouse. We could see that in the skin draining lymph node, and that being quite confined to that skin draining lymph node, but we didn't at that stage have the technology to be able to do the single cell sequencing and to map those TCRs.
Yeah, I just was curious. I wasn't aware of that, but I do know that, this having the accessibility to actual human data is really much more informative anyway. So it's really fantastic that you've moved into this model system.
Yeah, it's really exciting because what we really felt was when clinical trials are being monitored, there can be such a time lag between before you see a measurable decline in C-peptide, and at that point the damage is already done. That's quite often a large amount of a person's residual C-peptide and their residual insulin production that they've lost. So one of the big benefits of this, we think, is if we know the phenotypes of the cells at a given point, that we can very rapidly monitor people during the immunotherapies.
Yeah, no, it's a really, really great system. And we can't wait to see what you do next. We'll be reaching out to you regarding the TCR repository, should you have any interest in just even sitting in or joining us. The data you're generating is very exciting. So thank you again, Stephanie. Really have a great rest of the day, and thanks for all you're doing in this space.
Well, thank you ever so much for inviting me. It's been a real pleasure. Thank you.
I get a lot of well dones and wonderful talks just in the chat, just to have to say that. Okay, bye-bye.
Thank you. Bye.